Hello, everyone. Welcome to our monthly Science of Alt Protein seminar. My name is Bianca Dada, and I'm the Senior Scientific Partnerships Manager at the Good Food Institute. For those of you who are joining us for the first time, welcome to our community. The Good Food Institute is an international nonprofit organization that works to unlock the knowledge and resources needed to transform how meat is made, satisfying rising food demand in ways that accelerate climate progress, protect animals and ecosystems, and strengthen public health. In short, our goal is a thriving world fed sustainably. If you're interested in diving deeper into the science behind plant-based, fermentation, or cultivated meat technologies, I encourage you to explore our open access resources at gfi.org/science. With that, I'd like to introduce today's speaker. Professor Nesli Sozer is a leading food processing researcher at VTT Technical Research Center in Finland. Her work focuses on plant-based foods, sustainable protein production, and hybrid food systems that combine plant-based and cellular agriculture technologies to improve the taste, nutrition, and functionality of alternative proteins. She currently leads the Global Center for Food Innovation and Diversification to Advance the Bioeconomy, an international initiative focused on hybrid food innovations. Professor Sozer's patents have led to the creation of a recent spin-off company called Happy Plant Protein, which develops sustainable, high-quality plant protein solutions. She also coordinates the EU-funded Sustainabite project, which develops resource-efficient and minimally processed technologies for nutrient-dense plant-based foods, and leads the ProScale project, which focuses on scaling up single-cell protein production through continuous fermentation. With a career devoted to pioneering sustainable food innovations, we are delighted to have her joining us today to discuss how we can bridge functionality gaps through hybrid food design. Nesli, I'll turn the floor over to you.
Thank you very much, Bianca, and hi everyone in the virtual room. I cannot see you, but I guess we can keep the dialogue active after my presentation in the Q&A session. So I truly believe that the hybrid foods topic is becoming even more and more interesting. So I'm also happy to hear that there will be a follow-up presenter after this talk in the beginning of July. So that also gives the GFI point of view perspective that hybrid foods topic is really an enabler combining plant-based and also fermentation-made solutions. And actually, in the title, I have picked closing the functionality gaps. But when we talk about hybrid foods, the gaps are not only focusing on functionality. It is also there are other challenges beyond the functionality. So the scalability, especially when we look at the fermentation-made solutions, so the SALAG solutions, the cost, and not to mention the consumer familiarity. So in fact, the hybrid food perspective can tackle different areas of challenges and gaps. And I pretty much believe that most of the people in this audience would agree with me that plant-based foods have an important role in creating a more sustainable food system. We have also lots of strong scientific evidences, including the EAT Lancet reports, showing their potential in reducing the environmental impact and also moving towards more nutritious diets. However, we also know that sustainability itself alone does not drive so much the consumer buy-in. So consumers eat foods that taste great and fits to their budget and fits to their expectations. So if we want a larger scale adoption, plant-based foods must simultaneously deliver taste, nutrition, and cost, and balancing all these three simultaneously is rather challenging. So when we look at the plant-based and animal-based proteins, they are fundamentally and structurally different. They differ in molecular composition, structure, functionality, and behavior during processing. So as a result, when we are trying to find alternatives to animal-based proteins, it is not just swap one ingredient with the other one. It is much more beyond that. And we frequently encounter technofunctional challenges that relates to solubility, protein solubility, forming gelation, then nutritional challenges, presence of anti-nutritional factors, and as I mentioned already a couple of times, even with the plant-based alternatives, the consumer acceptance is oftentimes low. So during this seminar, I will be focusing on the meat challenge, because meat is when we look at the array of different animal source proteins, meat is the most problematic one from a sustainability and also nutrition perspective. And I pretty much like to show this slide because it surprises people. Many people think that plant-based meat is a recent innovation, which came actually during the COVID pandemic and post-pandemic, and it was the hype period, and people are not anymore interested about it. But as you see here, the plant-based meat existed more than a century. So this is a German advertisement, Flaschenfleisch, Obodo, which means simple as plant meat. And what is also interesting is that the marketing messages are almost identical to what we use today. Natural can fully replace meat, it's nutritious, it has 22% protein, it is half of the price of the meat, and it's tasty. So it pretty much resonates to today's modern plant-based meat, health, affordability, sustainability, and sensory quality, which was communicated 100 years ago. So this historical perspective reminds us that the challenge has never been simply inventing the plant-based meat. The real challenge is achieving the widespread consumer adoption and continuously improving the taste, because although this has existed more than a century, it's not still a mainstream product. So on the other hand, within this 100 years, we have advanced a lot in extrusion technology, both in dry extrusion, wet extrusion, supercritical extrusion, fermentation, precision fermentation, and so on and so forth. Then we can actually adopt these plant-based meat alternatives to next generation, where we can improve the taste, the price, and hopefully increase the consumer buy-in. So I would say that the question has never been plant-based meats. It's an innovative product and so on. I would say that that the plant-based meat is not a passing trend, and it has survived already for more than a century. So the only challenge we have to tackle today is how to make the next generation meat alternatives significantly better than the previous versions. Saying that, this picture actually shows a woman who is still biting on a animal meat-based burger. And she knows the sustainability and health-related pain points to red meat consumption, but she still continues to eat meat because it delivers a very attractive sensory experience for a large population of consumers. They love the appearance, they love the flavor, they love the texture. And current plant-based alternatives still struggle to reproduce all of these characteristics simultaneously. So we have to rethink our strategy in plant-based alternatives or meat alternatives in general. So are hybrid foods the solution? So what if we don't expect from one ingredient, in this case, a plant-based ingredient, oftentimes when we are talking with alternatives, to solve every challenge, techno-functional challenge, nutritional challenge, price challenge, and everything. What if we strategically combine the ingredients and technologies so that each contributes on its own strengths? So this idea indeed forms the basis of the hybrid food solution or hybrid food design. As a matter of fact, when I think about the future food system, I don't see a world dominated by a single production method. Instead, I see diversification. When we look at current diet, it's animal-based, but then also the plant-based side relies on 4 crops: wheat, corn, soybean, and rice, although we have lots of untapped opportunities also in the plant-based domain. Going forward, we will likely integrate plant-based and select ingredient solutions, and in some cases, I also believe that we will keep on consuming animal-derived protein-rich ingredients. So the hybrid foods is a practical pathway for combining these different resources and transitioning towards less animal-dependent diets. One key enabler to this desired future state is cellular agriculture, where instead of relying solely on animals and agricultural fields, we use cells as bioreactors and as production platforms. We can produce microbial biomass, animal cells, plant cells, but then also we can produce more targeted proteins, lipids, bioactive compounds, and other valuable ingredients. What I would like to highlight here is that oftentimes there is the threat that, okay, will the cell-ag approach totally tear apart the traditional agriculture? No, I don't think so. I believe that cellular agriculture will be only complementary to the traditional farming practices. Coming closer to hybrid foods. So there are such foods where we can combine bulk ingredients coming from animals, plant ingredients, or microbial biomass together with high-performance, low-inclusion ingredients that we obtain from precision fermentation or combining plant cells, animal cells, or more tailored ingredients which can be obtained by molecular farming. So the first example I will give is the simplest form of hybrid food design, which combines plant proteins with animal proteins. Although this may not represent the ultimate long-term solution for everyone, it is very useful from a scientific model because it helps us to understand which components of meat contribute most strongly to sensory quality and texture. And in other words, how much animal-derived material is actually needed to improve a plant-based food. So this is one of our published papers, which you can reach out from the DOI link in the right-hand corner. And it is a hybrid extrudate where we combined half and half minced beef, which has different fat ratios, 7% fat and 17% fat. And this is pretty much in Europe, a European level. 7% fat in a minced beef is a low fat, and 17% is a high fat beef product. And we combined it with pea protein isolate and texturized pea protein concentrate. The main ideology here was never to take a high-quality minced beef, but the idea was to utilize side streams which are microbiologically safe, but for some reason it doesn't fit to the quality expectations, and it's a side stream coming from animal-based production. It can be beef, it can be even fish. But for practical reasons, when we advanced during the project, we decided to make the proof of concept with a minced beef product. And we also looked into how the type of pea ingredient, if it's an isolate obtained by wet extraction or a texturized pea protein concentrate, how it affects the functionality. And in fact, what we have seen, and the picture on the lower end shows the TVP base, so the texturized pea protein based extrude. Top level is a pea protein isolate. It generates quite a distinct structure. And when we look at the textural properties, so the average compression force, samples with texturized pea protein, they have formed the fibrous structures with thin fibrils, which is also shown in the compression force. They had the highest, and we obtained lower compression force with the PPI containing samples. So when we look at the stereomicroscopy images, the top row shows the structural differences of the hybrid extrudates from the outer structure, and lower row is showing the inner structure after we bend the extrudates. The yellow arrows shows the flaky layered structure. So the isolate containing samples, they appear more crumbly and less structured, which was actually to our surprise. And what is interesting is that also to highlight that, of course, with the wet extraction, the PPI is already denaturated or partially denaturated. Then with the texturized protein, it is almost completely denaturated. And the common general knowledge is that during high moisture extrusion, the denaturation of the protein is affecting or dictating the protein structure. But what happens actually is that under shear flow process, the protein melts, the hot protein melt, and water mixture decomposes into two immiscible phases, described as spinodal phase separation in traditional polymer physics. And both the spinodal phase separation and thermodynamic incompatibility influences the formation of fibrous structures during high moisture extrusion. And understanding these mechanisms still remains one of the major scientific challenges in extrusion technology. Let's move forward with the sensory results, which is, I think, equally interesting, because when we have added the beef, minced beef, it has significantly reduced the characteristic pea flavor and increased meat-like sensory attributes. The samples containing texturized pea concentrate had much milder beany flavor profile compared to pea isolate, and more pronounced meaty odor and umami taste was observed. Also, according to the PCM model, we have seen that the short-chain fatty acids and the monounsaturated fatty acids were positively correlated with the meat-like other properties. And we also know from previous literature that especially the short-chain fatty acids and certain monounsaturated fatty acids, such as palmitoleic, stearic, and oleic acids, are indicators of indicator fatty acids for meaty flavor. Thus, having only a small fraction of these fatty acids are enough to bring the meaty taste. So overall, this work has showed us that it's possible to come over the undesired flavor and textural responses coming from the traditional plant-based ingredients. And then this brought us to another question. Can we achieve similar improvements without using animal ingredients at all? And this has motivated our next work, microbial lipids, to close the sensory gap in plant-based meat alternatives. So when we look at the meat composition, it's 70% water, 20% protein, and of course, depending on which cutlet of the meat you are looking at, it has roughly around 10% fat. And if we look at the research activities across the plant-based meat alternatives or meat alternatives in general, we have paid almost all our attention to the protein case, and we haven't worked much on the fat. And I think it shouldn't be like this, as we have seen from the previous case. Fat is pretty much associated with the flavor, juiciness, and mouthfeel. And microbes can, in fact, they are highly efficient cell factories for producing lipids, which can be tailored towards specific functionalities where they can deliver the expected sensory outcomes with improved sustainability and with better nutritional quality. So this is another way of looking at the hybrid food design, where plant-based proteins ingredients can bring in the structure textural component, and then microbial lipids can bring in the flavor, juiciness, and complement overall eating and cooking experience. When we look at microorganisms, not all of them can produce lipids equally. So most common yeast, the traditional baker's yeast, for example, Saccharomyces, can only accumulate little amount of fat, which is less than 10%. However, there's a specific group of yeast, they are called as oleaginous yeast. They can produce more than 50% of their dry mass lipid. The examples include Yarovia, Cryptococcus, Trichosporon, but also Rhodotorula, which comes with a bonus. As you see on the right-hand side lower corner, the nice orangish color. We can even go more close to red colors with specifically with Rhodotorula. The lipid yield, the composition can be tailored by controlling growth parameters, pH, temperature, oxygen, and specifically carbon to nitrogen ratio. So this is another work that we have done at VTT, and you can also download the publication from the QR code. And here our aim was to identify different microorganisms capable of producing food-grade lipids by using food industry side streams. And from the microorganisms that we have screened, we've identified Apiotrichum brassicae as a particularly promising candidate, which can produce a versatile array of fatty acid profiles. And after scaling up the production, we extracted the lipids using both enzymatic and ultrasonic pretreatments. We characterized the fatty acid profile, the distribution of triacylglycerols, melting curves, and so on and so forth. And we benchmarked it to the common lipids coming from plants or animal sources. And what we found is that our sample was high in palmitic, stearic, and oleic acid. Those were related to the meaty flavor. But then we also found that the tags that we have obtained were mostly monounsaturated or deunsaturated and showed pretty much similarity not only to beef fat, but also palm oil, which is another problematic oil that is oftentimes used in meat alternatives or animal alternatives in general. And we also found that specifically the melting curves, the third melting peak, resembles similarity to cocoa butter. So as a conclusion, the lipid produced by this particular organism, Apiotrichum brassicae, showed similarities to cocoa butter, palm oil, beef fat, so to say, problematic food lipids where there is either resourcing, sustainability, or health issues. And this is an ongoing work that's also part of our global center project, where we will be integrating these lipids, in fact, with plant-based meat alternatives, and we will do also sensory, we will check the sensory properties. Of course, sensory quality is not only determined by fat. When we talk about meat, color and flavor is also associated with heme proteins. And this is where precision fermentation opens another exciting opportunity. And this particular example focuses on heme proteins, where we can control both the color and flavor characteristics associated with meat. And in this work, we have used our trichoderma platform to produce hemoglobin-like proteins, which are basically derived from plants. And we benchmarked it with myoglobulin. And I'm pretty much sure you don't read the numbers, but that's not so important. So the highest peak, which is around 400 nanometer, reflects the heme binding. So the higher the peak, that means that the more heme is incorporated. And we see that the sample, which is labeled as HB1, has even higher heme binding peak than the benchmarking myoglobulin, versus when we look at the second sample, it has failed our expectations. So these were done in very small scale in 96 well plates, but then we scaled it up to 20 and 100 liter fermentation. And what we have seen on the right-hand side is that, in fact, the HB1 sample is quite similar to myoglobulin. And this is also another work that's part of the Food ID Global Center project. And we will be also in the coming weeks, months, continue the work and engaging the obtained heme to plant-based meat alternative setup. So far, I have discussed how hybrid food design can address the sensory gaps through use of microbial lipids and heme proteins. And this example is a little bit different. It's more looking at the texture and structure side. And here we are using mainly fungi as architects of structure. And we have combined extrusion processing, dry extrusion, with fungal solid state fermentation. And as you can notice, this part of our research is under patenting process. The process is quite straightforward. It starts with minimally processed ingredients. In this particular example, it's a whole grain oat flour combined with fava protein concentrate that's obtained with dry separation, and wheat bran. And the innovation, so the IP, is not only restricted to this set of ingredients. So we can expand it to different minimally processed different ingredient categories. First, we do dry extrusion to generate a scaffold for solid-state fermentation, and then we proceed with solid-state fermentation, fermentation time varying from 18 hours up to 30 hours or even longer. And this is how it looks. So you can easily see from these pictures how the fungal growth progresses over time. And after approximately 24 hours, the fungus has already colonized in most of the substrate. And at this point, we begin to observe a more cohesive structure and also a mild, nutty, and slightly meaty aroma. So this is particularly interesting because here we bring the texture component, but also we get the promising meaty and nutty aromas that is associated with meat-like taste profile. And the good thing here is that the starting ingredients, they already provide the high protein and source of fiber claims through use of these minimally processed ingredients. And this is a microstructure image of these meat alternatives, hybrid meat alternatives. And as you would see, the quantity of mycelia is clearly increasing when we move towards 24 and 30 hours of fermentation. But the light microscopy shows a better perspective on how the microstructure looks. And it gives further insights. So the fungal mycelia becomes progressively denser with increasing fermentation time. It goes in and around of those scaffolds, small extrudates. And at the same time, we saw also indications that the plant cell wall structures are getting modified. and also it transforms the proteins. And here the red color refers to protein, and then the turquoise refers to cell wall from the microbial biomass. And fermentation obviously also influences the nutritional quality. It has increased the free amino acids. This suggests that the proteins are becoming more bioaccessible. Although the drop in the phytic acid content, which restricts the bioavailability of the proteins, but also mineral bioavailability, it was statistically significant. And interestingly, and I don't have that results here, but I can speak it out, the in vitro protein digestibility did not change so much when we move from 18, 24, and 30 hours of fermentation. But what I can say is that we have benchmarked it with cooked chicken. It was almost identical. So we get a protein digestibility similar to cooked chicken breast. Then we looked at some basic technofunctional properties and textural properties, and extrusion increased the water binding capacity considerably compared with the original raw material mix. And during fermentation, the protein solubility gradually increased again. This is likely coming from the combination of fungal protein production and enzymatic breakdown of the existing plant proteins. And at the same time, we have seen an increase with the fermentation time on hardness and chewiness. These are all desirable characteristics when trying to create meat-like textures. But what has been most encouraging for me during this research project was that when we started cooking it at 200 degrees in a typical oven for 6 minutes, we didn't see much change in the thickness. But what we have seen, especially after 30 hours of fermentation, since the cell biomass, the biomass, fungal biomass has strong water binding capacity, it doesn't release it immediately. So the products were more juicy due to intense network of hyphae, which is of course very important from an eating experience. And I hope this video will work now. Let's see. So this is a video taken after cooking of the hybrid plant fungi meat alternative. And as you can see, the hyphae acts like a connective tissue, and you can also get a feeling of the juicy look. So just showing it once more. Now I will move to a very different approach, that is 3D food printing technology. And here we have combined pea protein with bacterial single-cell protein and used extrusion-based 3D printing to create meat-like structures. And just to briefly explain the concept, we start with a printable paste containing protein fibers, and this was a hydrated texturized vegetable protein that was actually a pea protein concentrate. And we use different kind of hydrocollates, formed a printing paste, and then picked a digital design. We have made the design ourselves so that we can control not only the external structure, but also the internal architecture. And then after the printing, we have also solidified further the printed shape by using calcium cross-linking. And this is another published paper from our research group. So this is how the pea protein fibers look after hydration. And the main reason that we have used a bacterial single-cell protein, of course, pea protein being a plant-based protein lacks some of the essential amino acids. Single-cell protein, bacterial one, has a complete amino acid profile, but also it's rich in micronutrients. It has high amount of iron and calcium as well. And we used alginate and alginate locus bean gum combination. And here you would also see details about the recipe of the printing paste. When we are talking about 3D printing based structuring, of course, the paste rheology is absolutely a critical parameter to obtain, related to shape fidelity. So the material needs to flow through the nozzle under pressure, but it must immediately regain its structure after deposition. So it must have mechanical rigidity and sufficient yield stress so that the printed object would simply not collapse. And the molding approach we have is that basically we have used the same printing paste and then simply molded it into a rectangular structure. So here we show the paste rheology, and based on the rheological data, the printed paste behaved like a soft solid, and that solid-like character is the one which allows the material to maintain its shape after the extrusion-based printing. So the storage modulus, which is G prime here, reflects the mechanical rigidity, so elastic properties. And then and this is quite critical, and this is what we need for layer by layer printing. And it's the highest when we combine when we have used the combined hydrocolloids, so alginate and LBG, locus bean gum. And then what we have seen, the yield stress as compared to alginate only sample, it is also higher. So this means that the combination of alginate locus bean gum improved the mechanical stability of the paste and made the printed structure more resistant to spreading or collapse after the deposition. And these pictures I like the most. Actually, these are taken by X-ray microtomography. And for some reason now you don't see it as it should be seen. It looks a little bit weird, just the green lines there. But anyhow, so those green pictures, the green shapes, they are referring to the pea protein fibrils, and then the grayish one refers to hydrocolloid single cell protein gel. And then the dark black color shows the air or void space. So from these X-ray images, we can see that the 3D printed samples, they yield a very organized fibril structuring. And then we don't see that organized structure when we simply mold the same printing paste. For the printed samples, cutting against the aligned structure, so the transversal direction required a higher force than cutting along, so the longitudinal direction. And the anisotropy index, which is also another important quality parameter when we are talking about meat and meat alternatives, it captures this difference as the ratio of transversal to longitudinal cutting force. And clearly the printed samples, they had higher anisotropy index than the molded samples. This is important because the actual animal-based meat itself is anisotropic and directionally structured. This final slide connects the microstructure to macroscopic behavior of the meat alternative. What you see is the 3D printed samples can be easily folded in the controlled direction, whereas the molded samples show lower flexural strength and are more prone to cracking. That difference is consistent with the earlier microscopy and cutting force results. So alignment creates a more cohesive and direction-dependent structure. And the broader takeaway is that extrusion-based 3D printing can be used to create a fibrous anisotropic meat alternative. However, the future work should really focus on generating high throughput outcomes, scaling up, and also optimizing the fibril and gel properties. And the last example is maybe the most futuristic one, where we combine microbial biomass with plant cell hybrids. So this is actually Arctic bramble, what you see in this picture, quite bright, nice red color. So how about we utilize the plant cells to bring the color component, the bioactive compounds. They also contribute to the structure of the plant-based hybrid meat alternative, and then utilize the fungal microorganisms to form the basic protein-based structure and help also elevate the nutritional value. Then we use the lipid-producing organisms to bring in the fat component. Before I move, I would like to introduce, probably this is not a familiar topic, the plant cell cultivation. It starts from the plant material, from the plant tissue, where cells are isolated and induced to form first a callus tissue. And from the callus culture, we can proceed to cultivating in suspension systems before scaling up to larger bioreactors. Compared to conventional agriculture, plant cell cultivation offers, similar to other cultivation techniques, several potential advantages. It's independent of seasons, climate conditions, quality is more consistent, and you can obtain highly valuable compounds in a controlled environment in a sustainable way. Plant cell cultivation is different than the cultured meat production. Plant cells don't need a scaffold and can be cultivated in suspensions, and they can contain up to 37% dietary fiber, 19% protein, and on top of that, valuable phenolics and bioactive compounds. And actually on the right-hand side, you see the actual food prototypes that we have done mainly by using plant cells. So they would still require some post-processing techniques to turn them into food and products. Oftentimes when I present plant cells, a question that I'm asked, how does it taste? How does it taste? And they can vary a lot in their sensory profiles. Some can resemble original plant material, but with much milder flavors. Some can be almost neutral, but this flexibility is actually advantageous because it gives additional formulation options. In other words, plant cells should not necessarily be viewed as copies of existing plants, but as entirely new ingredients. Okay, so what we did is that we have individually harvested these biomasses, microbial biomass and plant cells, and we followed a very simple process. We mixed the slurry, so the slurry contained 80% microbial biomass, which consisted of the fungal biomass and then the oleaginous yeast. We molded and pressed and fermented into a meat-like patty. Critical thing is the choice of the microbial biomass. And the reason is that here we used both trichoderma and rhizopus biomass. The left-hand side shows the trichoderma, which forms a very thin but more compact cohesive structure. And then on the right-hand side, you see rhizopus as the other filamentous fungi, where the structure is the hyphae is more grainy, much more thicker, and it forms a less cohesive structure. And there is obviously also the color difference. Trichoderma had lighter color than rhizopus. And in this picture, what you see is that on the top part, the mixture of the berry cells, so plant cells, together with fungal biomass that contains trichoderma, rhizae, and Yarrow lipolytica before cooking, and in the lower part is after cooking. And basically, the color is solely coming from the berry cells. And when we look at the delta E, which gives the color effect by inclusion of plant cells. It's much higher in the trichoderma sample simply due to the fact that trichoderma, starting from the beginning, has a lighter color, so you see more pronounced color change by adding the plant cells. And then when we look at the hardness, springiness, and chewiness, regardless of whether we add the plant cells or not, the rhizopus samples, because you remember the microscope images, they had thicker and more grainy structure, the hyphae structure, we obtained higher hardness, higher springiness, and higher chewiness values. So finally, I would like to say that our vision for hybrid foods is to accelerate the transition to a more sustainable food system by combining traditional and novel ingredients to familiarize also the consumer with this new way of food production, create new market opportunities also for novel fermentation-made ingredients. And I believe this approach will also unlock untapped new possibilities for innovation by bringing together the strengths of different food sources, but different expertise. So this was basically my last slide. So I would be glad to take questions.
Thank you so much for that incredible talk, Nesli. I am especially struck by kind of like this expansive view that you've taken of like looking at all of these different building blocks across nature, across different techniques. It's really inspiring, and that third project in particular is really getting my brain spinning a little bit about what's possible there. So thank you for that.
Thank
So
you.
cool to hear about it. Got a lot of questions in the chat, and I also have a couple of my own that I'd love to add in. So I wanted to start with just a high-level question about what challenges you see for hybrid foods that might be unique as opposed to the challenges that you see on projects that are specifically focused on plant-based or specifically focused on microbial fermentation. Like, what are the things that happen that are more difficult when we put all of these pieces together?
That's a very good question, and I would give a very straightforward answer to that. It's the labeling. Labeling is the biggest hurdle, I would say. So how to label, because especially when let's take the simplest case, plant-based and combining plant-based ingredients with animal-based. It's not an animal-based product. It's not a plant-based product. It's somewhere in between. But then I also give this example. Majority of those traditional patties, animal-based meatballs, they oftentimes contain at least 10% a plant-based ingredient just for binding properties.
Mm.
And people never, label those, or there could be some strategies that if we look at the dairy category, we have this term called half and a half. So the half and a half can be also utilized for meat alternatives. And especially I think in some of the at least the commercial products that are in Europe, this is an approach that is used at the moment. So they are using the half and a half terminology to say that, okay, it's a mixture of beans and peas together with the meat. But I think the labeling would be the largest hurdle. Also, this is not probably a major problem when we are looking at meat alternatives, but from the dairy alternatives perspective, it is not simply a mix and blend when we are combining plant-based with animal-based ingredients or with precision fermentation obtained ingredients. So there will be still some technological or processing related challenges that we have to come over.
Thank you for that. The labeling point is very interesting also because, um, you brought in this historical perspective, and it's so interesting to see, for these kind of legacy products, what's allowed versus the situation that happens when we start introducing new things. Yeah, thank you for that really comprehensive answer. We have quite a few audience questions, so I'm going to start with some of those. The first one's from Lucas. Can you please clarify which three side streams were used as biomass for the microbial lipid production in the examples that you gave?
I can broadly say, and of course you can check from the publication because that includes the amount of details that we can share. But it was a dairy side stream rich in lactose.
Great. Okay. And there was a question, kind of a follow-up on that, about whether brewer spent grain would be like a good example to use in a case like that, or if that's something that you didn't find to be as effective.
Uh, we didn't work with brewers spent grain for that particular case, but I can reply with my general experience there. Uh, each of these microorganisms, they have their own character. So,
Mm,
so it doesn't mean that any side stream they will eat up everything. There are some fungi which you can feed whatever you want to feed them. They will eat and consume. But then sometimes there are those inhibitors which actually restricts the growth of the microorganism. So if you tackle all those growth inhibitors and open up the structure so that the sugars are released, so really you can utilize other feedstocks than sugar. And in fact, the new project that Bianca mentioned briefly while she was introducing me, the ProScale project funded by the European Commission, we will be focusing on different large industrial side streams to provide alternative feedstocks to sugar. So that would be something that we will be looking at. And there are several different kind of lignocellulosic biomasses similar to brewer's spent grain.
it's a really big area of opportunity, I think, and I'm very much looking forward to hearing what you find in that study. But thank you for that answer. I'm going to combine two questions from the audience as a follow-up to that, from Mustafa and Monica, and they were asking a little bit more about the specifics of the main challenges and bottlenecks of introducing microbial biomass and its derivatives into food, which you started to get into, and then kind of even more specifically, what are the challenges of getting microbial fat to interact with protein like matrices in these hybrid products? And how can we make sure that we're selecting the suitable fat for these applications?
So the first question is the microbial biomass, and then the second one is the microbial lipid. Yeah, with the microbial biomass, again, it depends on which biomass we are talking about. Is it a bacteria? Is it a yeast? Is it a fungi? So there are several things. So we have to, of course, the downstream processing is very important in the functionality of the biomass. And again, in these presentations, I kept my focus on the meat challenge. And if I can boldly say, the incorporation of biomass in a meat alternative setup is easier than incorporating it in a dairy alternative due to the fact that oftentimes, again, this depends on the specific properties of the microbial biomass and then the downstream processing. But in general, a major complexity with the microbial biomass is the restricted protein solubility. And as you know, in majority of the food applications, protein solubility, foaming properties, gelling properties are quite critical. So I would say the technofunctional properties could be a limiting factor with the microbial biomass. So in the downstream processing, so far, majority of this biomass production has focused on reducing the nucleotide content.
Mm.
However, during the downstream processing, we can more efficiently, cost effectively do all these needed functionalization. Let it be enzymatic functionalization, or let it be some thermomechanical functionalization techniques. We can much more easily integrate as part of the downstream processing. Or otherwise it will be the pain point for the food formulation people. But this could be handled as part of the downstream processing. For the microbial lipids, actually that's an area which incorporating of microbial lipids to the plant-based proteins is something we are just now starting, and we are looking at different options there. Do we need to really extract everything, or do we keep them within because the lipids are accumulated inside the cells, so it's not secreted outside of the cell? Do we keep it inside, or do we encapsulate it with something else? So that's still a pending area of exploration for me, so I don't want to speculate much more there.
That's very fair. The insight about incorporating microbial biomass potentially being simpler for meat products than dairy is really fascinating because it's kind of goes against some of the conventional instincts that I might have or things that I hear where oftentimes people kind of pitch dairy products as being easier to achieve. And in some ways in the market, we're seeing so many plant-based dairy products out there. So it's very, very interesting to hear that and a really helpful thing to have highlighted. Um, and also your comments about kind of integrating upstream and downstream processing and making sure that yeah, we're not passing things on to the food formulators that are too difficult to work with is really well taken. And it's something that I think we all collectively need to think about a little bit more in terms of how to build those bridges and make sure that the things that are being produced are actually well designed for where they're going. So thank you for highlighting that as well. Um, got a question from Kevin along kind of similar lines about um, it seems to me that yeast and other microbes can already produce the components such as proteins and lipids. Why do we need the plants in alternative meat at all?
Diversification, one word answer. So the future is that should be diversified. It's already animal dominated, and then comes the plant-based. So in order to diversify our food production, in order to have more resilience and also as you know, this is no secret. Everyone knows that there is geopolitical stress everywhere around the globe. So in order to have access to food, whatever is the condition, so we have to have different access to different raw materials. So my simple answer will be we have to diversify the production, we have to diversify our raw material sources.
Yeah, we've been thinking that about that a lot in terms of resilience and like you mentioned about just being more impervious to environmental conditions, weather, all of that. So I think
Mm.
all of these factors are coming together in a really interesting way. And when you were talking about plant cell cultivation, it did make me wonder, outside of just this application of trying to create meat, I'm
Mm.
curious about how much interest there will be for plant cell cultivation in places where it's just really hard to grow other plants too. Um, that leads me to another question around that plant cultivation piece. One of the biggest questions we get with cultivated meat is about like the cost and efficiency, and the resources that go into producing meat in this way versus conventional animal agriculture. I'm curious kind of how that plays out for plant cultivation, and what the resources, efficiency, and cost are for something like that, and if that's something you see as a barrier.
Yeah, that's a very, very good question, and we have been working a lot with the plant cell domain, and we have even publications related to the technoeconomics, feasibility, and sustainability indicators of this kind of production. Of course, this is pretty much case dependent. In the cultivated meat, what you are producing is a commodity product, that is the meat. In the example that I gave you, it's a high performance, low inclusion ingredient. And another example I will give you that's not related to meat alternatives and so on, but for example, we are very much known that Finland is one of the heavy drinkers of coffee. And, and we have had several public visibility also coffee grown in the middle of Finland, so in our facilities by using plant cell cultures. But these are also premium products, so coffee. Then, of course, there has been also other groups who have been working on plant cells, and we know also successful startup companies working on cocoa production by using plant cells. But cocoa is again a premium product. And we have, of course, accessibility resourcing problems when it comes to cocoa or coffee. So it depends on for what purpose you are producing. But I think it's not a fair comparison because with the cultivated meat, it's a commodity product, and here it's a premium product or a high performance, low inclusion ingredient.
That's a super helpful perspective and brings me back to what you were saying about diversification. We just need to explore as many methods as possible to
Yeah.
see what will work in different regions. Two-part question from Primrose and Kevin, putting their questions together. The first part is, how is the sensory taste of hybrid products, and does that completely mask the beanie flavor? And the second part is, why do we need to make alternative proteins resemble meat flavor and texture? Do you think it's possible for consumers to get used to alternative proteins if their taste and textures are still different from meat, but they're still good?
Okay. I have already shown some of the sensory data. So basically, when we blended plant-based ingredients with the animal meat, so there was definitely tremendous increase in the umami flavor. I have actually quickly looked at the fungi biomass and then the extruded. So the extrusion first using scaffold and then moving to solid state fermentation, the sensory results. So it does also mask the faba bean concentrate, the dry fractionated faba bean concentrate. I don't know if there is someone in the audience who have tasted it, but I have been eating whatever. I'm like the guinea pig in the lab. It's challenging. I will put it politely. It's very challenging. And after this process, it really gets this nice umami taste after especially after the cooking. So yes, we have checked the sensory. We did not check though with the microbial lipids. As I said, that's an ongoing piece during the NSF Business Finland funded project, Global Center project. What was the other question? There
The
was one.
second one was about if you think it's possible for consumers to get used to alternative proteins, if their taste and texture is good but different than meat.
Yes, but this will take even longer time. I mean, changing people's behavior is extremely difficult. And actually, there was another project which Bianca, you have mentioned in the Sustainabite project that's also funded by the European Commission. We are looking at this solely from the consumer perspective. And in that project, we are not using any of those common knowledge, extrusion processing, and so on. We are using totally different kind of processing, very much focusing on minimal processing there. And if you look at, for example, falafel, as simple as that, nobody thinks that they are eating a meat ball alternative, but it is like that. And it's spread across nations, although it has been a traditional product. Everyone likes to eat it, and they don't think that they are replacing meat or this is a meat substitute. The texture is totally different. Yes, there can be these kind of products. The other example I would give is hummus or hummus. And that's like it could be a spread, cheese spread or a dairy spread. But again, people don't take it in that way. But I think that's even a more difficult hurdle. And if we look at the general consumer, yes, generally the majority of the consumer likes to eat meat because of the way it looks, the way it tastes, and if we want to gain the majority of the population, we have to come up with diversified solutions as well.
Yes, I echo that sentiment. And I think that nice historical piece that you brought in is a good reminder that even with trying to stick to the things that people are familiar with, it still takes a century. So I share your sentiment there. Um, I think you spoke about this a little bit in your presentation, but we have two audience questions. Um, from Gihan and Julia about the digestibility of kind of the final product of these hybrid products, and if that would pose an issue for people with any sensitivities.
Um, well, I will first answer the question related to digestibility because sensitivity is another thing, not necessarily related to digestibility. So actually, this was one of the, I would say, ongoing debates. Is the protein digestibility coming from these meat alternatives enough and comparable to the meat itself? Of course, it has higher digestibility. And I I answer to that question with different perspective. One is that uh, think about what you eat daily. Do you eat only meat or meat-like products? Our diet is diversified, so we don't rely on one particular food source in our daily diet. So if we are missing some component, we don't have to have from all the products that we eat the complete amino acid profile, the complete micronutrients. So we have to look at this in diet level. So that would be my very straightforward answer. But then by using the hybrid approach, for example, I gave you a combination of pea protein, which we know that it's lacking certain amino acids, essential amino acids. You can combine with bacterial single cell protein that includes all the needed amino acids, essential amino acids, plus micronutrients. Especially in Europe, we don't have a protein intake problem. We consume much more protein than we should be consuming, but we have a persistent micronutrient intake problem. We have iron deficiency. We have also deficiency in dietary fiber. So when we are looking at protein digestibility, we should also look at what the plant-based or the biomass is bringing together with it. It's the dietary fiber. And if I extrapolate it even further, now there is a lot of interest to GLP-1 companion foods, which means that the importance of protein together with dietary fiber is very important. So by using a hybrid food approach, you are easily able to tick the protein and dietary fiber boxes. Also, the plant-based itself is ticking these two boxes if we are using those minimally processed, so the concentrates instead of isolates. So my answer would be that protein digestibility, it's not that straightforward. Does it need to be that much similar? But I have given the example. It was very much similar to chicken, cooked chicken breast. Then the other question I would reply is the sensitivity. I would link it to allergenicity and so on. And for that, we need processing. And that's why I like to combine different processes. For example, extrusion processing with fermentation in order to reduce as much as possible the anti-nutritional factors, the allergens. The only way to do it, and I will open another, ongoing debate, uh, how much processing is needed. If we are talking about alternatives, we need processing, clear. I mean, we need to get rid of all these anti-nutritional factors, allergens, and so on, and that requires processing. Processing is good.
Yeah, I think that is indeed an ongoing debate, and I find that vision so compelling of taking like the best portions of each of these existing foods, each of these technologies, and yeah, I am constantly struck by in in the US, for instance, the marketing around protein versus fiber and how that aligns with people's current diets and things like that. So very helpful perspective there. I think there's a lot we could get into about that. Um, we got another question here from the audience. Based on your view of the sequence of fermentation and extrusion from the literature and your experience, um, what is your view on kind of fermenting the plant proteins first and then extruding them to improve fibrous texture formation, or can the reverse sequence also be useful?
Actually, we have done that and we have even published that. The thing there is that when you do fermentation, if it is oftentimes it's acidic fermentation, the protein solubility typically increases, and that's not good for extrusion.
Hmm.
So you have to shift the pH, and you would see that we have publications there in the literature. So after that, you can proceed with the extrusion. But there the problem is you have to first do the fermentation, and oftentimes this would be submerged fermentation. Then you have to dry it. You are adding a cost factor. The highest cost factor oftentimes in ingredient production is the drying. And then you put it to the extrusion and you add back again the water there. And then, it adds into the cost factor. However, when we start with dry extrusion, where there is limited water addition and it comes, and then you continue with solid state fermentation afterwards, it's we didn't do direct, energy consumption, but I would imagine that that would be much more effective from perspective of energy consumption, water consumption, and so on. But of course, you end up getting totally different structures, textures, experiences. If you use fermentation first and then continue with the extrusion versus first extrusion and then fermentation.
Yeah, absolutely. It's a really interesting space to play in, and it kind of makes me think a little bit about your last answer about processing, or like, you know, some processing is needed to make any food ingredient, and then even just the order of how you're doing that processing has such dramatic impacts on what you get out of it. So there's
Mm.
so much to play around with there, and so much to think about in terms of how processed it's considered to be, and fermentation as something that is potentially more culturally accepted, but is still technically a food process. All of these things are just
Indeed.
very complex and interesting. There's a follow-up question on that one. Um, Beyond extrusion, do you think shear cell structuring, free structuring, or other non-extrusion approaches could realistically produce meat-like fibrous textures, especially if fermentation is used to modify the protein substrate beforehand?
Yeah. Uh, I have not worked with those techniques, but I know there are different groups working on it. With the shear cell, especially Wageningen University have been working on it, and of course I have been following the literature and so on. Yes, that's one way of producing the meat-like structures. But personally, and our research group at VTT, we have not been looking into shear cell and free structuring. But of course, those are other ways of producing meat-like structures, just like also 3D printing. So
Hmm.
there are different ways. But that's not an area that my research has been focusing on.
Awesome, thank you. Got a question from Vishal. In your opinion, are strain selection and fermentation duration the major factors controlling whether fermentation improves or reduces functionality for plant-based meat analog development?
Uh, you can do a lot by playing with the growth conditions. One other strategy maybe I would like to mention is co-cultivation.
Mm.
So using a consortium of different organisms can help achieving the targeted sensory functionality, but also especially when you are working with the traditional fermentation, safety is quite important. You don't want to keep the pH so high because then it might raise some safety concerns. So by using different microorganisms, you can come over that hurdle. Yeah,
Yet another
but
form of diversification.
if it's precision fermentation, that's totally different. Uh, there are only, I would say, maybe very little you can do just by playing with the growth conditions. You have to do strain engineering. So
Hmm. Awesome. Yeah, continuing that theme of bringing together as many different elements as possible and leveraging many different types of organisms to make this happen. Um, what is your ideal kind of hybrid product? If you imagine kind of pulling, if everything worked on the technical side, what are the things that you want to pull together and what would you be able to achieve with that?
that is the toughest question, Bianca. It is like pick which one of your child you like the most. So, okay. Hmm. So actually, if I think about the short-term impact, I would pick the combining plant-based with animal-based ingredients side streams. Actually, we have a quite a successful startup company. This one is not from VTT, but we have been working on that field a lot. So which utilizes fish side streams, combines it with plant-based ingredients and turns them into fish balls. So salmon balls is quite popular in Finland, for example. So turning them into hybrid balls, and it tastes very nice, very good product. So for the short term, achieving the short term impact and getting over this consumer resilience towards this plant-based meat alternatives, I think my goal would be the low-hanging fruit is the plant and animal hybrid. But the second one I would pick is combining fermentation, so precision fermentation or biomass fermentation together with plant-based, because I know in the other side, in the cell egg domain, scalability and access to large enough infrastructure is a challenge. So in order to close that gap, again, to increase the consumer making the consumer getting familiar to these new solutions, I would pick the cell egg and plant-based hybrids.
Excellent. Well, I will stay tuned for more development on those beautiful future products. And
Yeah.
you've given us so much to think about there with this menu of options that you've laid out and the approach that you're taking to kind of drawing from the best of each of these different technologies available to us. So with that, I'm going to close our Q&A and thank you so much for joining us and for engaging with all of these nuanced questions and for laying out such a great vision for the future for us. And I also want to thank all of our audience members for posing these wonderful questions for us. So thank you, Nesli.
Thank you very much. Bye.
Awesome. And thanks, everyone. We hope to see you at the next Science of Alt Protein seminar. Thanks for joining us today and have a great rest of your day.